this week's Tuesday lunch seminar, where we invite internal speakers, mainly from our EEB department, to talk about their work in research. And today we have Dr. Joanna Rifkin, postdoctoral fellow in the EEB department, working with Professor Gina Bauckham. Joanna received their undergraduate degree in biology and classical studies in Amherst College in 2009. After some time off, they returned to graduate school in 2017 and earned their PhD in the university program in genetics and genomics at Duke University.
I started grad school in 2011. I finished in 2017.
Perfect. So Joanna then was a postdoctoral fellow at the University of Toronto, focused on bioinformatics. And from 21 to 22 was lead geneticist at a startup, ISBA Environmental Laboratories, and before coming to Michigan. Joanna's research interests are in plant genetics and bioinformatics, and they especially enjoy developing genomic resources for expanding access to non-model systems and porting bioinformatics pipelines into creative environments. And at Michigan, they're working on projects with Dane's rocket and the common morning glory at the Bauckham lab. And today we will hear about recombination and the evolution of sex chromosomes in Rumex hastatulus. And after the talk, we kindly ask that the first question is from graduate students and postdoctoral fellows prior to the faculty questions.
Thank you for that detailed introduction. So yeah, hi, I'm Joanna. It's really exciting to be here today. I've been having such a fun time visiting the department and visiting Ann Arbor. This is the first research trip I've gotten to go on since the pandemic, so it's really fun. So today I'm going to be telling you a couple of different stories about the relationship between sex chromosomes and recombination in a cute little plant. I'm going to front load. Oh, it was working. Yeah.
Yeah, click on the PowerPoint and then try again.
That? Yes. Excellent. I'm going to front load my acknowledgements because it really takes a village to get any of these things to work. So here at UMich, I wanted to work with Gina for a long time. I was super excited to get the chance to do so, and it's just been a total treat getting to know the Bauckham lab and the department. At the University of Toronto, a lot of people contributed to the work I'll be talking about today. So I'd particularly like to thank my lab mates Fee Baudry, Bahul Chowdhury, Zoe Humphreys, Bianca Sacchi. Pasi Rastus is not in my lab. He's a fantastic guy though. And Stephen and Spencer ran the show. So I'm going to be giving you a little bit of background on how sex chromosomes evolve, particularly for everybody who's in ecology and doesn't think about chromosome structures and recombination most of the time. Then I'm going to be telling you 3 stories about recombination and chromosomes in Rumex hastatulus. I'll be returning to this outline periodically so that you can orient yourself if you happen to have nodded off after a big lunch. So sex chromosomes are really widespread, and we're used to mammalian sex chromosomes like these, but they're actually much more diverse than that. So everything on the left side of the screen here has sex chromosomes, which are pictured on the right side of the screen. Sex chromosomes come in different flavors. There are XY sex chromosomes, like these Rumex hastatulus ones here. There are also ZW, so in many species, including strawberries, many butterflies, and many birds, it's actually the female rather than the male that is heterogametic. So here we can see a ZW pair. There are even sex chromosomes in species that spend most of their lives as haploid, like this moss. Just to make things confusing, we call those UV, because we just sort of started moving around the alphabet for some reason. So I'm showing you all of these pictures of sex chromosomes and demonstrating that they're really morphologically differentiated. That's not actually what matters about sex chromosomes. They don't have to be different sizes or shapes. What makes sex chromosomes is that they have suppressed recombination and sex-determining regions. So this is a very old picture of recombination, just in case your memories of recombination go back a long way. Recombination, as you all probably know, is the process of crossing over that happens during gametogenesis, when chromosome segments are reshuffled. So the questions that we'll be circling around all have to do with how and why this recombination between sex chromosomes evolve. I'm going to be outlining a classic model of how we think this happens. So the basic model is that sexual antagonism creates strata. I'll explain all of these words. So you start off with a pair of autosomes like these. They're just regular chromosomes. They recombine over their whole length. There's nothing special about them, until one of them acquires a sex-determining locus. This locus does something to differentiate males and females. Now it's a sex chromosome. After some time, a mutation emerges that has sex-specific effects. Let's imagine that this is a plant, so maybe it makes males flower earlier. This locus has a sex-specific variant, so you want the early flowering form in a male and the late flowering form in a female, because otherwise, well, that's maladaptive and you're in trouble. So any variant that's going to reduce recombination between these two loci, that'll have an advantage in spread. Over time, the lack of recombination between these segments will allow other changes to creep in, like maybe this will get more heterochromatin and more pseudogenes. And then it happens again. And over time, this area of suppressed recombination creeps up the chromosome. This isn't just a theoretical model. We have evidence for it from a variety of systems. We can make a prediction that these strata that evolved at different points should have different degrees of divergence. And indeed, when we look in nature at sex chromosomes, that's exactly what we see. So on the right, you have a human sex chromosome, and you have very old strata, a very old stratum that's very diverged, and a very new stratum that's not diverged. In a totally different part of the tree of life, through convergent evolution, exactly the same thing has happened here in Silene. This is a powerful model because it makes really specific spatial predictions about many aspects of the genome. For example, how much recombination should be happening between these chromosomes where, how effective selection should be given the effective recombination rate, and where you should have genes that are and are not expressed. Rumex is a great system to look at these questions because it has a lot of diversity for all these kinds of traits. So across the genus, you have variation in whether plants are dioecious or not, how many chromosomes they have, how many of those chromosomes are sex chromosomes, and you have multiple origins of sex chromosomes. One of the wonderful things about studying sex chromosomes in plants is that many of the mammals you're familiar with basically share the exact same set of sex chromosomes. Cats and dogs and mice, it's all the same sex chromosomes. Not so with plants. We have multiple origins, convergent evolution, X, Y, Z, W, it's wonderful. So hopefully I've given you enough background that you can follow the next story I'm going to tell you, which is taking a look at how these sex chromosomes have evolved. So I'm going to zoom in on one species of Rumex. This is already published. If you want more detail, I can send you the paper. This species is very weird. In the western half of its range, it has 4 autosomes and an XY pair. So far, so familiar. In the eastern half of its range, though, it has 3 autosomes, 2 Ys, and a giant X. Somehow this is a thing that the plant has decided it wanted to do. We don't judge. What's wonderful about this as an experimental system, though, is that we can compare an old sex chromosome with a new sex chromosome, and we can compare a sex chromosome with the autosome it used to be. This is great for testing these predictions that I told you about, where we expect different things to be happening spatially depending on how old the sex chromosome is. So the data I had to work on this were a brand new linkage map and a brand new genome assembly. At this point, the genome assembly was in only about 7,000 pieces, which was pretty great for a plant. We also had some pedigree and population sequencing data from earlier papers that we could realign to this new genome. I'm going to be leading you through many complex figures in this talk, for the which I apologize, but that's how the data are. So this is the first of them. And the layout is going to be really straightforward, but there's going to be different data in different lines. So up here, we have 5 chromosomes. Chromosomes 1 through 4 and the XY. Along here, we have different sets of sequencing data. The pedigree data, a linkage map, population association mapping data, and population genotype presence absence data that are consistent with XY. The main thing I want you to focus on is the one of these things is not like the others. So what you should be looking for is similar patterns along the autosomes, a different pattern along the X, and between these two cytotypes, sometimes this is going to look like an autosome, and sometimes it's going to look like a sex chromosome. Is everyone braced for this? Okay, so what's exciting about this enormous heap of points to me is that indeed, sometimes this looks like an autosome, it's gray. Sometimes it looks like a sex chromosome, it's blue. So this is looking at inheritance patterns in a pedigreed cross. And in the cytotype with 2 sex chromosomes, this is acting like a sex chromosome, but here it's not. Similarly, in the linkage map, here this is showing an association with sex, if you just QTL map for sex. Here it's not. And in the cytotype with 2 sex chromosomes, these actually come out as 1 chromosome. So the linkage data is consistent with that fusion that we know about from cytology. Same thing in the population data, and same thing in effectively a population GWAS. This chromosome comes out as sex-linked here, both of them come out as sex-linked in the blue. Okay, so far so good. We've found our sex chromosomes. What I'm going to show you next is a series of plots on describing recombination rates. So this is what is called a Murray map. It's named after a French person who worked on timetables for trains, which is a fun fact. And this is a way of describing recombination spatially along a chromosome. So in both of these maps, on the y-axis you have something that describes recombination. Here you simply have the position in centimorgans. Here you can think of this as the derivative of that slope, so how steep it is. The things I want you to notice about this Arabidopsis map are we have this steady pattern of up, 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 plateaus at the middle, up, up, up. That's reflected in the derivative. So there's no recombination around centromeres. The other thing I want you to notice is the scale. This is 30 megabases. That's an extremely large bacterial chromosome, but not a very large plant chromosome. So we expect recombination to act really differently between the sex chromosomes and the autosomes. But when we take a look at the comparison of the sex chromosome and the new sex chromosome, we don't see as much of a difference as we expect. So here in the X, which is shared, they both recombine at the ends and are flat in the middle. Here, in the new sex chromosome, when it's a sex chromosome, it recombines at the ends and is flat in the middle. When it's not a sex chromosome, it recombines at the end and is flat in the middle. So even though the linkage positions of these are different, the extent of recombination between these chromosomes is not very different, regardless of whether it's a sex chromosome or an autosome. That's a little weird. What about the rest of the genome? So if you think about that Arabidopsis picture I showed you, there was steady recombination all the way along. Here there's these massive flat sections. So this is again in megabases. These are hundreds of megabases with no recombination happening at all. That's a little freaky, given that our model posits that recombination happens everywhere until it becomes a sex chromosome. The implications of this for sex chromosome evolution are really interesting. So I showed you this model at the beginning, that sex chromosomes gradually lose recombination along their length. This seems to suggest that instead we're in a world where there is limited recombination everywhere, and the sex-determining region just kind of popped in this low recombination region. I'm not the first person to think of this. There's actually been some really interesting modeling work on whether it's easier for sexual antagonism to evolve if there's less recombination. And it probably is. So what I have here are different recombination rates and the region between them where the polymorphism is maintained. The less frequently a new locus recombines with the sex-determining region, the easier it is for it to invade. So what's cool about that is it might actually be easier for sex chromosomes to form in this pre-existing recombination suppression. In plants generally, this is that Arabidopsis I showed you. This is it to scale next to this Rumex map I showed you. And even though this is our model plant that we pretend is a normal plant and like to study, this is much more representative of what plants are like. So this is a barley chromosome. And what's interesting is even though barley doesn't have sex chromosomes, it does have this massive non-recombining section, just like Rumex. If we look across plants and ask whether the sex-determining regions are more likely to turn up in suppressed recombination regions, that seems like it probably happens. We're still fairly early days in figuring this out, but both sex-determining regions and also self-incompatibility loci that have some similar evolutionary patterns tend to show up in enteromeric regions. What's exciting about this to us is that this shows us that in Rumex we have a standard model of how we think sex chromosomes evolve, and something different is happening in plants with large genomes with suppressed recombination. So that model still has explanatory power, but it's limited for this taxon. So that's an exciting finding, and we can move on to our next section. So the next story I'm going to be telling you is about sex differences in recombination. All of the recombination I told you about in part 1 was sex averaged. However, there are more differences in male and female recombination than just in sex chromosomes. I'm going to give you some examples. So this is the example that is most familiar to all of us, because we all took introductory genetics, and we learned that males in Drosophila don't recombine. And we were like, that's weird and terrible. Why are fruit flies like that? And the fruit fly biologists just said, don't worry about it. It makes fruit fly biology easier. We love it. So this isn't super common, but it's also not limited to fruit flies. So this is a fruit fly. Males don't recombine. This is a silk moth. Silk moths are ZW rather than XY. Females don't recombine. This is a worm. This is a lily, which is really odd because it's not XY or ZW. It's a hermaphrodite. It makes both ovules and pollen. But when it makes pollen, the pollen doesn't recombine. Achiasme is a really extreme case of sex differences in recombination. However, sex differences in recombination that aren't that extreme are really common. So this is across a very, very, very broad range of life. And if you look at this distribution of recombination rate estimates, there's a real skew just across everything, where females recombine more often than males. I want to draw attention to this because, again, the range of sex determination systems across these species is super wide. We have XY, ZW, we have haplodiploids, we have hermaphrodites, and yet in almost everything, except marsupials, because those are so weird, females recombine more than males. Often not significantly, but still often. So we can go one step further in comparing male and female recombination rates and look at where it's happening along the chromosome, not just how often it's happening. So this is a graph averaged across all human chromosomes of where along the chromosome, relative to the centromere, recombination happens. And you see this really striking pattern, that male recombination happens at the tips, and female recombination is, compared to males, skewed towards the centromere. Okay, fine, humans are boring. But we see the same thing in sticklebacks, which is, I think these are guppies, which are slightly more exciting. And again, female recombination all along the chromosome, male recombination just at the tips. We can go one step further and look across a lot of animals and a plant. And again, this plant doesn't have separate sexes. And you see this really widely convergently evolved pattern that male recombination is tip-focused, female recombination is centromere-focused. That's very weird. When we started working on this, there was no data yet for dioecious plants. So we started out with 4 questions about what is happening here in dioecious plants. So I'll be circling back to all 4 of these questions, but basically, given what we already know about how recombination is unusual in these species, how does sex-specific recombination and the distribution of recombination along the chromosome fit into this pattern? So the data we had for this, we had a new and improved linkage map that actually differentiated male and female recombination. I could explain how, but no one wants to hear it. And we had a new and better genome assembly. So for that first question, do we see heterochiasmy? Is there just a basic difference where females recombine more? And to ask that question, we're going to compare map lengths. So how many centimorgans are there? Because number of centimorgans reflects number of recombination events. So what we have here is the sex averaged map from part 1, the male map, and the female map. And there are 2 things I want you to notice about this table. The first thing is that, yes, across all of the chromosomes, the female map is longer, the male map is shorter, and the sex average map is somewhere in between. This is good. It means we were maybe measuring something real the first time. The other thing that I want you to see is that there are these big stretches with just no crossover events at all. And finally, the sex chromosome is an outlier. Particularly this non-recombining section in males is massive. But in terms of these differences and the extent of non-recombining sections in both sexes, it's not that big an outlier. Again, recombination is really infrequent in this species. So if you remember the Murray maps, which I drilled into your head in part 1, we're going back to Murray maps now. So at the top, we have that same Murray map with centimorgan position and megabase position. This time males are purple and females are green. And at the bottom, rather than the derivative of the slope, we have the difference in crossover number. So how many more times we have crossover events in males versus females. So what you can see is there's kind of a mixed pattern. There's a lot of areas where there's big differences in recombination. It's not so simple as tip-focused, though. It's kind of, I mean, A1 is that very nice tip-focused pattern, but A2 we have a few recombination hotspots, and they aren't the same between the sexes. Here in the sex chromosome, we have a region in males that does not recombine, that is the size of an entire Arabidopsis genome. It's upsettingly big. So if we circle back to our first 3 questions about landscape dimorphism, we do see heterochiasmy, we do see more non-recombining in males, and we think that this might have facilitated the evolution of sex chromosomes because, remember in part 1, I told you it's easier for sexual antagonism to invade when there's not recombination. So if it's easier for sexual antagonism to invade non-recombining regions, and the non-recombining regions are concentrated in males, that suggests that XY rather than ZW will evolve. And that's the pattern both across the tree of life generally and in this species. So now I'm going to be asking why this happens. And I'm going to present 2 models. There are other models in play, but these are the most interesting ones to us right now. So the first reason this could be happening is haploid selection in pollen. The pollen gametophyte, the pollen is a gametophyte for those who don't do plant biology. You can think of a pollen effectively as a miniature plant that is doing many functions of a full-size plant, but it only has 1 copy of its genome. The main thing that it's trying to do, among its full spectrum of biological functions, is grow successfully down a style to an ovule. So there's a few different mechanisms by which selection during the gametophyte life stage could lead to reduced recombination in males. So maybe selection is acting to suppress recombination in males because of pollen. Alternatively, maybe the important stuff is happening in females. Maybe there's selection in females to increase recombination and push recombination towards the centromere. And the way that would be working is that meiotic drive often focuses on the centromere, so anything that increases recombination near the centromere could counteract meiotic drive. So these are the two models that we explored in the most detail. And I'm going to show you predictions that would be evidence for these two models. So to do this, we sliced up the genome into regions and looked at correlates of recombination in different regions. We think that if haploid selection in males is what really matters, then the more male distortion there is, the better something is at getting passed on in pollen, the lower the recombination rate will be around that. Alternatively, if there's distortion in females, we predicted that would positively correlate with the recombination rate. So there would be an association between areas where distortion was happening and selection for recombination to increase. So far, so simple. Except those aren't the only things happening in the genome. In general, genome content correlates with the recombination rate. We don't entirely understand why. There's a few ideas, but what I'm showing you here is genes in black, and then RNA and DNA TEs. So there are TEs that live in low recombination regions, and TEs that live in high recombination regions. So when we slice up our genome and look for correlations, unfortunately, there's a lot of other things that could also explain what's happening. So now that I've thoroughly beaten you over the head with models, let's get into some data. If you look across the genome, distortion varies. So this is how much bias we have in transmission for male versus female alleles. And in general, there are many more sites that are significantly distorted in females. But it really varies, and it varies a lot across the different chromosomes and by position. So whatever's happening in this genome, it's actually happening really heterogeneously, and different things are happening in different places. If we look at those correlations in slicing up the genome, the first thing you can see is that everything correlates with genes per window. Number of genes you have is a major predictor for everything else that's happening in the genome. If we just zoom in on the relationship between distortion and recombination rate, though, it's actually consistent with both of our predictions. And I'm going to make that a little clearer by giving you now the same correlations, except I've taken out the genes. I've just controlled for the genes. This is everything that's left after number of genes. And there it's even a little clearer. We see this negative correlation between male crossovers and male distortion. And we see a positive correlation, though very, very faint, between female crossovers and female distortion. And we see the biggest difference between crossovers in distorted, in regions with low male distortion. So overall, this is kind of consistent with the prediction for both haploid selection and meiotic drive, but everything in here is kind of, all the effects are weak because these were really complicated linear models and everything in a genome is kind of autocorrelated. So it's kind of some tantalizing and interesting circumstantial evidence on why these recombination landscape differences evolve. But I think there's a lot more to do here because it's this incredibly widespread pattern. And it's so unusual that you see the same thing happening across just so much of life. So that's the most complicated part. And now we're going back to one more story about how genomes evolve, and then there will be time for questions. So the last thing I'm going to be telling you about today is not published yet. It's pretty new and pretty hot off the presses. Well, hot to the presses. And this is stuff that Mark Hibbins is working on as a postdoc at the University of Toronto now. So I told you at the beginning that Rumex is a really good system for this stuff because it's really diverse. I'm not expecting you to take in these entire phylogenies. I'm going to simplify them. But the main take-home is that we've had a 2005 ITS phylogeny and a bigger and more detailed recent chloroplast phylogeny. They mostly agree with each other. This species is in a different place. But you can see here the scale of variation across the genus. So I'm going to start here. Most species in the genus, about 150 or so of the 200 species, are hermaphroditic. They either have perfect flowers or they're monoecious. So for the animal people, they either have flowers that have both male and female reproductive organs, or they have male and female flowers, but they keep them on the same plant. So the vast majority of them, yeah, they're all about the same. They have a base number of 10 chromosomes. There's diploids, tetraploids, hexaploids, all hermaphrodites, all 10 chromosomes. Then you start moving into this part of the genus, and things get a lot weirder. You start to see real changes in the chromosome numbers. In here, they get down to 8. This group all has 7 base number of chromosomes with an XYY system. This group, which is where hastatulus is, the one I told you about earlier, has between 4 and 8 chromosomes, an XY system, an XYY system. Our best evidence suggests that these are totally different sex chromosome systems. So whatever happened, like here, it's weird. The genomes just went wild. So we thought, okay, well, we know there's a history of genome rearrangements. Maybe we can figure out what it is. Maybe if we sequence a species in this part of the genus, we'll be able to understand what happened from here to here. Maybe it'll be like this beautiful, beautiful diagram from Arabidopsis. So down here we have Arabidopsis lyrata. It has 8 chromosomes. Here we have our beloved Arabidopsis thaliana. It has 5 chromosomes, all of which together are smaller than a single Rumex chromosome, but that's okay. And we can actually trace all of these different translocation events. We can see how these chromosomes got rearranged. Great, awesome, we have a plan. So to look at the relationship between these two genomes, and the Rumex fulicifolius genome is super new, I'm going to show you a synteny dot plot. So this is a sample synteny dot plot of humans and chimpanzees. What we have here are chromosomes, other chromosomes, and shared sections between the chromosomes. What you can see is that sometimes things have been moved from chromosome to chromosome, sometimes they've been turned backwards between the two species, and sometimes they're basically identical. So those are Arabidopsis chromosomes I just showed you. Let's look at them in this perspective. So it's a little more complicated, but let's take lyrata chromosome 1. We can see that indeed most of it is collinear with our Arabidopsis chromosome 1, but then there's also this section here, which is Thaliana chromosome 1, which got moved over there. We can see there should be some of lyrata 2 lining up with Thaliana 1. Great. Okay. So now let's take a look at our new fulicifolius and hastatulus chromosomes. We'll see what segments went where. It's going to be great. It's not great. So this did not live up to my wildest expectations of being able to trace what happened. But there are some really interesting things going on. First of all, we can find some synthetic regions, but also we can find an awful lot of reshuffling, particularly in the middle sections of these giant non-recombining chromosomes. So there's something cool going on here, where you have these giant non-recombining chromosomes, and they seem to just be able to make like chromosome soup out of themselves. And probably the way they're doing that is, if you're a large non-recombining plant chromosome, you are full of transposable elements. So you have so many copies of the same sequence. And that means that they can recombine with that TE anywhere else in the entire genome. So there's some really cool stuff going on in this group of species with large non-recombining regions, chromosomal rearrangements, and sex chromosomes evolving. So to get you back to our outline, hopefully I've told you some things about how sex chromosomes evolve, some ways that interact with recombination, and the way that can evolve across genomes. And we are in plenty of time for questions.
Gina, the first question is from...
Oh, I'm sorry.
Yeah, we need a grad student first. Come on, the grad students are shy in this department.
Ask about the name system. Okay, I'm just going to ask this question to warm everyone up. Okay, so that dot plot at the end,
Yeah.
lack of synteny was amazing. My question is in two parts, not because I want to dominate, but because I don't understand. So one is, first, how far apart phylogenetic, when did those two species diverge, do you know? And then the other question.
I do not, a while.
Okay, yeah, it would have to be a while, right? Like longer than the human chimp diverge?
I think so.
Okay, and then the next question is, have you all dated the LTR elements in either of the species? So to me that suggests, if you think there's a lot of movement going on because of the TEs, that there should be active TEs maybe in each genome, and so maybe you could get an idea of when they discovered it. Or is it relatively recent?
We haven't, that's a really good idea, particularly since, so people might not know, but you can date the age of TE insertions by particularly how many nested ones there are. So particularly that'll be, we have a lot of transcriptome data for hastatulus, so we can look at what's getting expressed as well as just what's there in the genome. The solicifolius genome, that just got done this summer. So we have a couple of pollen transcriptomes and flower transcriptomes for solicifolius. So I think that's something that I should yell at Mark over Slack to do.
But my question is about the non-recombining nature of these non-recombining regions.
Yeah.
You said they're kind of non-recombining, but that would be a large portion of the genome. So how non-recombining are they?
So one way we get data on how non-recombining they are exactly is if I go way back to the comparison with the population data and the, yes, but I want to make this big now. No, not like that. So if I go back, I'm just going to leave this in the... Okay. Yes? No? I think I might need help from one of the IT people. I think I broke it.
To get back to your presentation.
Yeah. Well, anyway, if you look at the comparison between the population data and the cross data, great. So what's nice about the population data is that that's capturing a lot more recombination events because this is effectively a GWAS down here, whereas the linkage map is just one generation's worth of recombination. So what's cool about this is we can actually pick up in finer resolution things like where recombination tails off in the sex chromosome. So here it's quite recombining there and then just tails off. Here, more association. And like what part is and isn't associated with sex. So if we compare the population data to one generation's worth of cross, it's still very non-recombining. Recombination happens very infrequently in these regions. It probably happens, but it's probably really, really, really infrequent and not that much more frequent on the autosomes than the sex chromosomes, which are totally non-recombining. Yeah?
For those images, it looks like there's a data point, essentially at every location, every X value along the position axis for all of the chromosomes. And I noticed that throughout the presentation, except for one of the earlier slides, I think chromosomes 1 and 4, there were like big chunks where there weren't any data points for some of the chromosomes or some locations.
Oh yeah, I think if I give you this one?
That one?
Yeah.
I haven't done genetics in a long time, so I just want to see this.
Yeah, so this is reflecting problems with our ability to relate the linkage map to the assembly. And everything in this section was done with an assembly from the XY cytotype, not the XYY cytotype. So pretty much what we were doing was taking mapped positions on the assembly and plotting their recombination positions. But particularly with the XYY cytotype, there were a lot of things that we just didn't have markers for or couldn't place. And that probably reflects rearrangements between the two cytotypes. And there's now an assembly for the other cytotype. So actually remapping the linkage data to that would probably fill in those gaps.
Where were the plants, what habitat were the genetic material?
All of this, so this was all done in the greenhouse. In the wild, they live in sort of sandy, scrubby habitat. And for the mapping, we actually did transcriptome sequencing for everything in the linkage map, which was intense, but we knew we were dealing with this big, messy genome. And in terms of thinking about ways of reducing representation of the genetic data, we thought, well, the thing about transcriptome sequencing is that's letting the plant do the selection for you and give you markers nicely spaced out. So another aspect of these gaps is I showed you there's a relationship between genes and recombination. So we had fewer markers in the non-recombining regions because they're comparatively gene poor. Though, I mean, I shouldn't exaggerate, there's a lot of genes in the non-recombining regions too. And like, I don't remember the percentage, but there's a solid chunk of the actual expressed functional genome that's stuck in these low recombination regions, and therefore subject to much less effective selection. Yeah.
That was really fun, and plants freak me out. I had a question about, so have you looked in these large non-recombining regions at diversity, like genetic diversity within compared to between populations? And the reason that I ask is it seems like it strikes me as being funky or potentially a bad strategy to have such quick evolution of sex chromosomes. It would lead to fast reproductive isolation between isolated populations and be able to do like driving stuff within each population. And I'm just curious about like, yeah, how that seems to shake out if you see reduced diversity within populations in these long non-recombining regions.
This is one of those questions where the answer to it is in a paper that I know I'm on and I don't remember it. I'll get back to you.
Okay, deal.
Yeah.
I don't know if I missed it, and I'm sorry if I did. But so the mechanism for this lack of recombination, are there like inversions, or do you know what is the reason why this large area of the lack of recombination exists?
We don't know. We absolutely don't know. So there are some inversions you can see between the two cytotypes here. These are inversions. But that's not, those are actually in high recombination regions. This one over here too. The most likely guess we have is that the recombination machinery nests at the telomeres. So recombination is telomere initiated. So our best guess is that recombination starts at the telomeres and reaches into the chromosome. And the chromosomes here are so big that the recombination can't reach. That's a very anthropomorphic answer. I'd like more questions from grad students. I know you folks have good ideas. Yeah?
I think this might have just been something I missed, but maybe end the talk when you compared the two species. What kind of mating system does Salicifolia have?
I don't think I mentioned. So Salicifolius is hermaphroditic. It's supposed to have perfect flowers, but it seems like it might actually be monoecious. We don't know a lot about selfing in this taxon. They're wind pollinated, so it's been very hard to determine what is and isn't self-compatible. We know that Salicifolius is self-compatible because if you put it in a bag before it starts flowering, you still get seeds, which is a very high-tech method of determining that. We have evidence that another weedy species in the hermaphroditic 10 chromosomes block is selfing from genetic data. So probably self-compatible. But yeah, that's comparing something way down in the 10 chromosomes, everything is a very normal plant range, to something in the weird range. Yeah?
How do you reconcile this repression of combination that you saw within a species, but with all of these really large rearrangements? Compare the two.
So I think for the large rearrangements, there's a couple different answers to that. So these large rearrangements probably happen through ectopic recombination. So recombination between different chromosomes, which is possible because they have matching TE sequences. That doesn't necessarily have to happen very often an event doesn't have to happen often to still happen in evolutionary time. So I would guess that they're rare ectopic recombination events, possibly because you have a section of TE that's comparatively close to a telomere that ends up close to one that's far from a telomere in a different chromosome. A lot of that would hinge on the spatial arrangement and positioning of chromosomes in the nucleus, which some people are doing really cool work on, and those people are not me. The other answer to that is there was a really provocative paper from the 80s in another species of Rumex, Rumex acetosa, which is familiar, tasty garden sorrel. You put it on fish. And that is XYY. So females have 14 chromosomes, males have 15. The XYY system there likely came from a fission rather than fusion event, but we're not really sure. But this team in the 80s went out and just did chromosome squashes across this population. And what they found is there was a huge amount of variability. So you'd have a lot of really different chromosome conformations just in this population. So whatever the mechanism is, it seems to be happening frequently. And they found evidence that those rearrangements seem to drive. So honestly, it's just very weird. And this is weird stuff that's happening in plants with big genomes, which are a pain in the neck to study.
Do you think that the suppression of recombination might be due to selection, like in avoiding recombination between TE-rich areas to avoid that ectopic recombination?
So certainly there's a lot of suppression of TEs generally, right? It's like TEs are methylated to keep them from doing their TE thing. I'm not up on the relationship between methylation and TEs, and recombination, but that seems like the sort of thing that could really turn into a feedback loop, right? Like you start accumulating TEs, so you start suppressing everything, and then there's less recombination, and then more TEs can invade. Can I try any more questions out of you, or does everyone want to get out of here? Great. Thank you so much for your attention. You know where to find me.